purpose. And what I will be telling you is a little bit following up on what Anita just talked about, which is an integrated view. If only my slide will move forward. I'm sorry for that. There we go. An integrated view of different aspects of aging. So we're not studying aging specifically. We're very interested in age-related diseases, particularly those that are associated neurodegenerative syndromes that are age-dependent or age-related. And of course, we're looking at things from the point of view of mitochondrial dysfunction, and also from the point of view of deregulated nutrient signaling, so deregulated metabolism. And as as you know, conceptually, you can look at a problem in two broadly different ways, if you will. One of them is, as we did in the 50s and in the 60s, to carefully dissect every component of the system and focus in one particular component and study it in depth, isolated from all the other components in the system. And the other way is to look at the whole system as a whole and try to understand how that system behaves in the environment that we are in. Of course, this is particularly important in the context of aging, because if each of the components ages at a different speed, but the components are connected to each other, we can expect that the result is not going to be very pleasant. So we're interested in these, and we're interested in understanding how mitochondria, through the interactions with other cellular components, affects aging and age-related processes. And we're stepping away from the reductionist views on mitochondrial signaling, because it's very clear for the past 20 years or so that mitochondria are not isolated from the rest of the cell. They have physical contact sites with the ER that are fundamental for lipid synthesis and calcium homeostasis. They share metabolic pathways, antiviral signaling, membrane deformation proteins with the peroxisomes. They are needed for the formation of peroxisomes. Lysosomes are necessary for the removal of damaged or excessive mitochondrial peroxisomes. Mitochondria release vesicle traffic directed specifically either to the lysosomes or to the peroxisomes, and the contents of these vesicles are different. And finally, mitochondria form contact sites with the lysosomes. So, and this is a summary of the, let's say, social interactions between mitochondria and other cellular components. So we can no longer just imagine a system in which there's nothing but mitochondria and nuclear gene expression, and mitochondria modify that nuclear gene expression, and that causes something else. We think it's really important to take into account different components of the cell and how they affect and are affected by perturbations in mitochondria. For instance, one of these perturbations is how mitochondria regulate lysosomal function. We and others have shown that under acute mitochondrial stress, there is a hyperactivation of AMPK signaling that is well characterized as a response to the energetic stress caused by a sudden defect in mitochondrial energy metabolism. This AMPK hyperactivation will trigger not just an increase in autophagy, but also an increase in lysosomal function and an increase in lysosomal biogenesis. This process goes on for a few hours, but at some point the cells decide that enough is enough, and they stop this program, and they actually enter a completely different program in which they shut down AMPK, which results in perturbation of lysosomal function, impairment of lysosomal biogenesis, and essentially the cells stop degrading dysfunctional mitochondria and with both dysfunctional mitochondria and dysfunctional lysosomes. So this is just an example of how mitochondria interact with other cellular components and can, through those interactions, have damaging or not damaging or protective effects that are long-ranging. So my lab has been interested in this question in how mitochondria and lysosomes affect each other. We usually depart from defect in one of these organelles, and see what happens to the other. And one of the things we've came to realize in the last few years is that whenever there is a defect in one of these organelles, there is a secondary defect in the other one. So there's no such a thing, at least from what we've observed, in which we have a mitochondrial defect or a lysosomal defect. We have mitochondrial and lysosomal defects. And having that in mind, we asked one question that we were curious about, which is what happens when the lysosomes cannot acidify. It's been shown in different model systems that impaired lysosomal acidification is more and more prevalent as organisms age. And we were curious to understand what are the biological consequences of these. Of course, the pivotal characteristic of the lysosome is that it's acidic. So the inability to acidify its lumen for sure would have some consequence, and we were interested in exploring that. To address that question, we took an approach that we often take, which is to generate a transcriptional of the perturbation we are interested in studying, in this case, inhibition of the VATPase, and from that transcriptional signature, identify which transcription factors are predicted to be hyperactive and which transcription factors to be repressed. And by identifying the transcription factors, we can then work our way upstream the pathways to try to understand what is the proximal and distal events that regulate those signaling pathways. So in this case, we were interested in understanding how VATPase inhibition affects the cells. We started, again, as we often do, using publicly available transcriptome data. In this case, it was 3 experiments done by different labs with different cell types, but they all treated the cells with bisphenol A1, which is an established inhibitor of lysosomal VATPase. So we analyzed the transcriptomes of these cells. We defined the differentially expressed gene list. We defined the transcription factors that were upregulated, hyperactive or repressed in each of these data sets. And we picked up the transcription factors that were behaving exactly the same way in the 3 different data sets. Of course, some of you will be thinking that this is a very dirty approach to the question. And you're right, it's 3 experiments that were not done by us and were done by someone else in different cell types. On the other hand, the fact that we can secure the common response from 3 different cell types made by 3 different labs confers a degree of reproducibility to the data. So these transcription factors that behave similarly in all 3 data sets yielded a list of 8, 2 of them associated with cholesterol synthesis and lipid synthesis, MEEK, P53, hypoxia-inducible factor 1 alpha and 2 alpha, which is also known as EPAS1. And in order to have some idea of how to prioritize these, we took advantage of that pathway analysis and builds on the known relationships between these proteins. We fed this list of 8 transcription factors to this program, and the pathways that seem to be prioritized have to do with response to oxygen levels and height. So that tells us that HIF1 alpha and HIF2 alpha are predominantly upstream of the rest of these transcription factors. So bearing that in mind, we abandoned we closed the transcriptional data analysis and we went to the bench. We got wild-type mouse embryonic fibroblasts, which is a model that we often use in the lab, and we treated them with baphyllomycin or salife, which are 2 inhibitors of the VATPase, of the lysosomal VATPase, that act by different mechanisms. So the hope here was that by doing the same perturbation in 2 completely different manners, we try to tease out to weed out any possible artifacts due to side effects of the drugs. And clearly we could see an upregulation of the levels of HIF1 alpha. You don't see anything in the control cells because that's exactly how it works. HIF1 alpha is made constitutively, and it's degraded. So HIF1 alpha is made, translated, the protein is hydroxylated by a group of enzymes called prolyl hydroxylases, which belong to the alpha-ketoglutarate superfamily. When hydroxylated, another protein, which is a ubiquitin ligase, PVHL, binds to it and targets it for proteasomal degradation. So all the time, degraded all the time. That's why you don't see it in control cells, even if we grossly expose the Western blots. So, and here we use deferoxamine as a positive control because it chelates iron, which is a necessary cofactor for the prolyl hydroxylase reaction. So it's a way that it's a good positive control to generate increased levels of HIF1 alpha. Of course, there's many reasons why HIF1 alpha can be increased when we inhibit the lysosomal PAs. One of them could be because there's decreased levels of these VHL protein. That was not the case. It could be because there was excessive levels of succinate, which is a product of the reaction, or of other metabolites that inhibit the prolyl hydroxylase reaction. We measured it. It was also not the case. There was no defect in alpha-ketoglutarate. We could eliminate that by measuring the levels of these metabolites. Obviously, the other two cofactors in the reaction are oxygen and iron 2. It cannot be oxygen because the cells were all in the same incubator, so they all have access to the same concentration of oxygen, which left us with iron 2. So we were a little puzzled. Why would impaired lysosomal acidification affect the levels of iron 2 in the cytoplasm? But then we started digging more into it, and it turns out that the levels of the homeostasis of iron is regulated in the cytoplasm. And when there is not enough iron 2, the cell mobilizes its reserves of iron, which are essentially ferritin, where iron is stored, and mitochondria because of the richness in iron sulfur compounds, and targets them for degradation via the autophagy pathway. So under iron starvation, mitochondria and ferritin are put inside autophagosomes and fused with lysosomes. In the lysosome, iron 3 is liberated through from its associated proteins and other atoms, and it's reduced to iron 2 by an enzyme called STIP3. As a good lysosomal enzyme, it works at a very acidic pH between 4.5 and 5.5. Iron 2 can then be released from the lysosome to the cytoplasm via two transporters. Doesn't matter now the name. The bottom line and the important information is that iron 3 cannot be released from the lysosomes. Only iron 2 can be put out. So our question here was, if we inhibit the lysosomal acidification, do we accumulate iron inside the lysosomes? We purified the lysosomes with from cells treated with baphyllomycin and, of course, vehicle control treated cells, using an immunoprecipitation approach, and lo and behold, we could find a clear increase in the amount of iron inside these lysosomes. So this tells us that indeed, if we block the VATPase, we can get increased iron storage in the lysosome. We then sought to determine, we already knew there was an apparent decrease in the availability of iron for the prolyl hydroxylase reaction, but we wanted to see if it's specific for that reaction or if it's a general iron homeostasis defect. So iron homeostasis is regulated by IRP1 and IRP2 in the cytoplasm, iron regulatory proteins 1 and 2. And under iron deficiency, they stimulate the expression of transferrin receptor, and they repress the expression of the ferritin's heavy and light chain. So in other words, they stimulate the uptake of extracellular iron and they repress its storage so that they can use it. We measured what happens when we treat the cells with baphyllomycin or salife, and there is a clear increase in transferrin reception. In transferrin receptor expression. You can see that the scale is different. Usually when we do baphyllomycin and salife, and we did the experiments in parallel, we get very similar effect, but the magnitude is always a bit higher in the baphyllomycin treated cells. we then sought to try to rescue this system. So we gave iron citrate to the cell medium, because iron citrate is not bound to transferrin, and can be, the iron atoms can be imported through the plasma membrane directly, so bypassing the lysosomal system. When we do, we can see a clear reduction in the expression of transferrin, as if by giving iron that can bypass the lysosomal pathway, we can rescue, so to speak, the iron deficiency phenotype. So we used other ways to show that there was functional iron deficiency, and that we could rescue it by providing iron citrate to the cells. I'm running a little behind, so I'll just speed up. We could see also that when we treat the cells with iron citrate, HIF1 alpha goes back down, and the expression of its targets also go back down. Because we were treating the cells with compounds, even though it's two different VATPase inhibitors working by a different mechanism, we were worried that we are still not immune to possible artifacts or side effects of these compounds. So we decided to take a little bit more genetic approach, in which we knock down one subunit of the VATPase, in the case the subunit V1H. We used siRNA for these. We used multiple ones. We picked up the ones that worked best. As you can see, the knockdown of the subunit is fairly efficient. And when we knock down the VATPase, of course, the lysosomes cannot acidify properly. I don't have the data here, but we made it. We have immediately a clear accumulation of HIF1 alpha, down regulation of ferritins, heavy and light chain, and increased expression of transferrin receptors. So by blocking the VATPase function through a genetic approach, we again obtain a functional iron deficiency phenotype. We then were curious to see if this happens also in real life, not in a system in which we wipe out one subunit of the VATPase. So we started looking through lysosomal storage diseases. Many of them are known to have dysfunctional lysosomes. Well, they are all known to have dysfunctional lysosomes, but many of them are known to have lysosomes that cannot acidify properly. So we focused on Pompe disease, which is a glycogen storage disease. The enzyme that is mutated, or in this case missing because we use the knockout mouse, is the enzyme called alpha acid glucosidase, which is necessary for debranching glycogen lysosomes. So these cells are known to have a large proportion of lysosomes that cannot acidify properly. So it was a perfect model for us to check if a model of physiopathological model of impaired lysosomal acidification was also resulting in iron deficiency. And indeed, we observed an increase in the expression of transferrin receptor, downregulation of ferritin, and increased levels of HIF1 alpha. So again, impaired lysosomal acidification results in functional iron deficiency. Of course, having these results in cell culture prompted us to test these in vivo, because we worry that when we find something in cells, that it's somehow an artifact of cell culture. So we started looking in vivo. But before I go to the in vivo data, just a brief word. I don't have time to go through all the details, but of course, we were curious to see if there's not enough iron in the cytoplasm, what happens to mitochondria, because mitochondria are one of the biggest consumers of iron in the cell. And this is a probe that reacts with iron 2 and is targeted to mitochondria. You can see that in the control cells, the mitochondrial network is very visible, but in the cells treated with baphyllomycin, so VATPase inhibition, we could barely see iron inside mitochondria. When we rescue the cells with iron citrate, again, we can see the mitochondrial network. This result is just a technical control, but it also tells us that mitochondria can easily import iron from the cytoplasm independently of the endolysosomal pathway, at least when they are profoundly iron starved. And of course, having iron perturbs the steady state levels of the respiratory chain complexes, perturbs the function of the respiratory chain, and results in a sharp increase in the levels of superoxide. And all these can be rescued by addition of extracellular iron that works through independently of the lysosomes. I will skip this slide in the interest of the story. So we started from the Pompe disease cells using MEFs from the knockout mice that models the disease. And we decided to look at what happens to the actual mouse, to the brain tissues, because as I said, we are quite interested in the brain, in the role of lysosomes in the brain. So we're looking at the cortex at 6 months and 12 months of age. Again, decreased levels of ferritin, increased levels of transferrin receptor, increased levels of IF1 alpha, so the data fits. We went one step ahead and measured the levels of iron 2 and iron 3. And as you can appreciate here in the knockout, at 6 months of age in the cortex, we had less iron 2 and more iron 3, which is consistent with the data that I showed you before, that the lysosomes, by not acidifying, cannot convert iron 3 in iron 2. We verified this result by in vivo MRI, but I don't have time to walk you through those details. Again, the mitochondria not working well. Here we cannot make the same type of experiments that we did in intact cells, but isolated mitochondria, we see that the complexes and the supercomplexes were not forming properly. And finally, we could ask the question, what is the biological consequence of impaired lysosomal acidification in vivo? By checking by taking the brains of these mice. Of course, this is not a pure response to impaired lysosomal acidification, because there's also the question of the glycogen storage and other functions of the lysosome that also get affected. But this is all part, as if we look at the system in an holistic manner, this is all part of the phenotype triggered by impaired lysosomal acidification. So in order to have an unbiased response, we did transcript of the 12-sold brain from the knockout and wild type, the cortex. We, of course, did pathway analysis. Many of the pathways that are highlighted here in red were very rich in innate immune signaling genes and proteins. And the most astonishing result came actually from the transcription factor analysis, in which the top 3 transcription factors are all interferon-related 7, interferon-related 3, and STAT4 are all deeply related with interferon signaling and sterile innate immune responses, what is usually referred to as inflammation. So having this result at 12 months in the cortex, we were curious to see, so just a brief word, these mice have predominantly a cardiomyopathy phenotype. So the brain is not supposed to be the primary tissue affected. So in that sense, it was ideal, because what happens to the brain because of the lysosomal defect without having to deal with massive disease in that tissue. So we saw that there was a very strong innate immune response in the brain at 12 months of age. And we decided to look at earlier time points to see from when does this inflammation signature start. We did that by qPCR. Of course, we got the same results at 12 months of age, a sharp increase in many of the interferon-related genes, but we also got the same result at 6 months of age and even at 2 months of age. There's already a robust inflammatory signature. So since we could rescue the iron deficiency by giving iron that does not need to go through the lysosomal pathway, iron citrate, we had the naive idea of doing the same experiment with mice and increasing the levels. Oh, I'm running super late, so I'm going to summarize the finding. We gave extra iron in the diet to the mice, and we could ablate the inflammatory signaling at 2 months of age. We could ablate the levels of the cytokines that were being produced by the mice. Just so you know, these are different cytokines that are triggered by this inflammatory response. The pattern is always the same. This is the wild type mice in the control diet, wild type mice in iron enriched diet, knockout mouse in the control diet. You can see a sharp increase in the levels of the cytokines, and knockout mouse treated with the iron enriched diet ablates the inflammatory response. This is very briefly that we think the trigger for this inflammatory response has to do with the instability of mitochondrial DNA, but we did not, we are still working on that, and we did not finish that part of the story. So to finalize, defects in the lysosome that can manifest as storage of iron 3 or in other defects, sphingomyelin, cholesterol, and many other components that should be degraded by functional lysosomes, result in decreased mitochondrial biogenesis and mitochondrial respiratory chain capacity, increased superoxide production, and inflammatory signaling. So we think that the interplay between mitochondria and lysosomes and between mitochondrial dysfunction and the deregulation of nutrient signaling can be a driver for many other of the known hallmarks of aging, like cellular senescence, genomic instability, and telomere attrition. Telomerase is also an alpha-ketoglutarase dependent superfamily, epigenetic alterations. As Anita showed you before, the proteostasis is intimately regulated, associated with the regulation of metabolism. So we think that by understanding better the crosstalk between mitochondria and lysosomes, we can also have a better insight how the different programs of intracellular and extracellular aging are associated with each other. Just a brief word to thank my funding from the European Research Council and from a patient foundation that funds research in Pompe disease. Virtually all this work was done by a fabulous student turned postdoc that I had in the lab, King Faisal Yambire, and acknowledge the superb collaboration by Philip West in Texas and Esther Meron Holtz in Israel. Thank you very much for your attention, and I apologize that I went over time.